Good morning, everyone. I'm Mark McClellan from the Brookings Institution's Center for Health Policy, our program on value and innovation in healthcare. I'd like to welcome you this morning to our public conference on improving productivity in pharmaceutical research and development, focusing on the role of clinical pharmacology and experimental medicine. We'd like to thank all of you for joining today. We know this is a crowded room here. We've also got a lot of people joining us online this morning. The interest in this conference, I think, is a reflection of the strong support for finding better ways to improve product development that many of you all have been involved with, I know have followed along with very closely. The conference is being hosted as part of Brookings' ongoing cooperative agreement with the U.S. Food and Drug Administration and in collaboration with the International Consortium for Innovation and Quality in Pharmaceutical Development. And it's been a real pleasure working with both of these organizations over the past several months in collaboration to develop the materials for this conference. You'll see from the agenda get our slides right. You see from the agenda that we've got a lot of ground to cover, and we hope also plenty of time for discussion, what we hope will be an insightful meeting on a very timely topic. Over the past decade, there has been widespread concern about the rising costs and reduced productivity in drug development with potential adverse impacts on patient access to much-needed treatments. Despite tremendous progress in our understanding of the molecular basis of diseases, of the translation of medical discoveries into safe and effective treatments, all this process remains a relatively slow, expensive, and high failure rate process. The high failure rate of investigational compounds, particularly in later, more resource-intensive stages of development around the so-called stage 3 clinical trials and the like, is widely seen as a driver of cost and lost opportunity for improved productivity in the whole research and development enterprise. Each of these failures also represents a lost or delayed opportunity to develop other promising compounds because of the higher resources needed to bring each of these compounds to the market or to determine that they're not going to work. So the focus of today's conference is on steps that are being taken through clinical pharmacology to change that. Focuses on the tools and methods of clinical pharmacology and how better, more systematic application of such tools could improve early-stage sponsor decision-making and overall productivity in the research and development enterprise. This is a very timely occasion for discussing such improvements. Public-private partnerships and pre-competitive consortia are demonstrating that it is possible to make a difference in improving the drug development process through sharing data models and other insights into particular disease areas. Policy and legislative proposals now being considered by Congress are aimed directly at strengthening basic and regulatory science and supporting a more efficient and effective development process. They're working their way through Congress right now. The FDA is undertaking a number of initiatives in these areas, and our understanding of the drivers and mechanisms underlying human disease continues to improve at a rapid pace. So more opportunities exist than ever before to work together to use clinical pharmacology to improve the way in which innovative drugs and biologics are discovered, developed, and approved for patient use. And we're looking forward to discussing all of them today. I'd now like to turn this discussion over to my colleague Greg Daniel for an overview of the day and some logistical issues for the conference, and then we'll get started. Greg, thank you.
Okay, thanks, Mark. I'd like to echo Mark's comments and welcoming all of you here today. It's certainly going to be a very interesting discussion in a room nicely sized for all of us in here. We can get to know each other a little bit better. When we first began discussing this meeting with our colleagues at FDA and the International Consortium on Innovation and Quality in Pharmaceutical Development, for the rest of the day we're going to use IQ as the abbreviation, we wanted to build an agenda that tackled the key challenges companies face in early-stage decision-making. And as such, our agenda for the day is structured around these challenges and the role that clinical pharmacology can play in addressing them. Session 1 will be focused on target identification and selection. Session 2 will center on the issue of dose optimization and strategies for improving dosing studies. Session 3 will tackle issues of clinical trial enrichment, precision medicine, and the process for ensuring that investigational compounds are active in the right patients. And then finally, Session 4 will pull together threads from all of these previous 3 panels to explore the better use of early clinical pharmacology studies and how they could improve the prospective design of later stage trials and contribute to the totality of evidence approach for demonstrating efficacy. Discussion during these sessions will inform a closed-door roundtable with staff from Brookings, FDA, and IQ that will lay out potential next steps for improving the use of clinical pharmacology tools and the ones that we discussed today. It's expected that this conference will lead to subsequent roundtable discussions. What we talk about today can inform potentially future FDA guidances, establish best practices for the application of emerging clinical pharmacology tools, and potentially to support academic publications. We have a great lineup of speakers with us today, and we're really excited to get started. Before we begin the day, I'd like to just go over a few specific housekeeping items. As you'll know, the agenda is quite packed. We have a lot of panelists who have come with even more slides to go over today. So to our specific panelists, I just ask you to make sure that you try to remain within the allotted time that we've given to each of the speakers. For all of your ease and enjoyment, Ahimsa is down in front to let you know how many minutes you have. So please pay attention to the numbered cards as they get closer and closer to zero. Each of the sessions, we do have allotted time for active discussion as well as questions and discussion with all of you in the room. During the panels, we will have microphones. They're right now in the back, but we'll bring them up so that if you do have questions during the panels, we will make sure that we get to all of you with those questions. We are webcasting this event here today live. And for those of you who are watching via webcast, the slides should be viewable in real time, but there is also a PDF packet, I think, on that link that you've logged into the webcast that you can also download those slides. As you may have seen, we do have coffee and beverages in the room. It's, I guess you go that way, and it's the next room over with all of the coffee. Lunch is on your own, but there will be a food cart open here at the hotel for your use. There are also lots of restaurants in the area, and there are maps and a list at the registration desk out front. And then finally, this conference, this webcast, is being recorded, and an archived video will be available online on the Brookings website. With that, it's now my pleasure to introduce our first 2 keynotes for the morning, who will be helping set the stage for the rest of the discussion. We'll first hear from Dr. Janet Woodcock, the Director of the Center for Drug Evaluation and Research at the U.S. Food and Drug Administration. Dr. Woodcock will be immediately followed by Dr. Mark Rogi, Global Head of Clinical Pharmacology Early Development at Biogen. With that, Dr. Woodcock, I'll turn it over to you.
Good morning, everyone. I don't have slides because I just have a brief talk, and there are many slides to follow. So we're here to discuss, I think, one of the central problems in drug development, which is there's a huge pressure to reduce the costs and improve speed and efficiency of drug development. And there's no doubt, and many future speakers are going to allude to this, that the failure rate, the lack of predictability, is one of the central drivers in drug development, that so many compounds fail, especially in clinical development. And so the question is, what can be done to reduce that? Now, we have sort of 2 competing models that we've sort of been running over the years, if you'll forgive me for saying this. One is the relentless sort of checkbox-driven march through phase 1 and 2, okay? Let's get to phase 3, all right, and let's get a dose, let's demonstrate safety, check, and let's get into those efficacy trials. The problem is about half of those trials fail, and they're very, very expensive failures. And we've known this, everyone has known this for a very long time. But the competing model, the question is how viable is it to spend more time to really learn early in development, preclinical development, and during early development so that you improve your probability in phase 3. And that's really a hypothesis that's going to be discussed in the next 2 days. Can you really do that, and how well can you do that? And I would propose that between the first time we started discussing this, which was 20, 30 years ago, and now, there have been tremendous scientific advances. So that hypothesis is more likely to be true now, okay, than it was in the past. And the question is, are we at a stage where we can really apply modern tools, modern understanding, and get that success rate up? And clinical pharmacology, of course, is the central discipline to bridge between the basic scientific hypotheses and what you're going to go into phase 3 with, and how you're going to do that. And the question is, how much can we use that discipline? Can we apply it better? Can we use a more holistic model and really improve our success overall, and even actually have better doses? Bob Temple here sitting in the front row, he claims that we almost never get the right dose when a drug is approved. There's so much uncertainty still about the dose that can we do better than that? So the product that we get out on the market is actually a better product as well. And I think between improvements in mechanistic understanding, biological understanding, and then computational capacity, it's worth a good bet that we could do better. And I think many examples will be shown in the next couple days about that. Now, there are 4 themes to this meeting. The first is, can we get target selection and target validation right early on, and what can be done to improve that? Now, there's been a tremendous difficulty, really, in understanding whether or not you're trying to impact the right target. And much of that is scientific gaps. So, for example, the NIH project, the AMP project that's going on with the industry in looking at targets for some major diseases is an example. That's a very comprehensive program, which is basically goes through the basic science and into the clinic and back in an iterative kind of learning loop to see, can we distinguish those targets that really are that really would, be worth developing drugs to. And pursuing targets that have been validated in natural experiments, okay, for example, GWAS studies, or where we've done replacement therapies for various things, we have a much higher success rate there. It's been claimed in literature, okay, because we already have that natural understanding of the role of that particular enzyme, mechanism, whatever it might be, in the pathophysiology of disease. And so can we do this without those experiments? Can we understand better through our own clinical pharmacology experiments, really, and holistic integration of knowledge, which targets are worthwhile pursuing? So we have to do quantitative analysis of drug targets, the networks they're embedded in, understand that better, and the effect of drugs on targets and networks to the extent we can. And we need to integrate, of course, the animal models, not looking at animal studies as simply check toxicology, 2-week toxicology, rats and dogs or mice or what have you, but really thinking about these are experiments that can yield insights into whether or not what we're pursuing is what we should pursue. And what also, speaking of networks, what are the off-target effects? Because, of course, toxicity is one of the big failure reasons later on in development. And so I think integration between the animal, the non-clinical studies, and the early clinical studies has to be one of the hallmarks of this effort. So something that can unify this is what we're developing as models. And these models go from the understanding of the target and binding and so forth all the way through to the whole animal and the natural history of disease and what's known about it integrated into one model. And, of course, this is fantastically complex when you get into the details, but at least we're not sort of blindly intervening and just waiting and experimenting with thousands of people to see if actually our predictions are true. So that's one big area. Can we move to a different model, at least where there's enough biological and pathophysiological understanding to construct those models and to test compounds within those models? And I think that'll be part of a discussion, the first discussion. And then selecting and optimizing drug dosing before confirmatory trials are conducted. Now, as I said, it's long been recognized that dose selection and regimen design is essential but neglected, right, and not done adequately, or not done in a way that would optimize the drug effect and minimize the side effects. So you can do, though, fit-for-purpose trials that by design allow for exploration of a wide range of doses and therapeutic use contexts like you can do titration designs and so forth if you design the trials correctly. And somebody's going to put in their slides, and I have to refute this, of course you don't have to do pairwise comparisons, and that's the only way to figure out what the right dose is. Of course you can look at exposure-response relationships, right? And I would say, as the head of CDER, if anybody, CDER tells you can't do that, talk to me, because what we want you to do is explore the widest range of doses possible and use the mathematical modeling and so forth to understand the exposure response and dose exposure response curves. So, but again, the empiricism was, one of the reasons people haven't been able to do that is they've only tested 2 doses in phase 2, right? And so, then you're going to be doing a pairwise comparison, basically. So, we'd like to see a dynamic range, and we'd like to see that. But again, this is new type of, not that new, but this is newer types of ways of doing the exploration of the exposure response space. The next one is studying the right patients. Now, we're making progress in this. More than a decade ago, we began putting in place the regulatory processes, the policies, the infrastructure to support the challenges of regulating what personalized or precision medicine products. So this did not catch us unawares. And this trend, I understand we've approved quite a few, even this month. We've approved quite a number of targeted therapies, and we only expect this to increase over time as there's greater biologic understanding. We have issued guidance on enrichment, and we really endorse enrichment, but enrichment is only good if you understand why you're doing it, basically. And the more biology and quantitative understanding you have behind that, I think the better off you are. Obviously, selecting the right dose is extremely important. And in the dose exposure response space, exploration of that space, we can also understand in early studies how intrinsic factors, intrinsic to the patient, other intrinsic factors such as drug metabolism and so forth, impact and cause variability. And knowing that, I think before you move forward, how much variability in response are you going to have, if you can possibly measure that, is extremely important. And then supporting demonstration of efficacy. The classic, of course, and what we usually ask people to do is to randomize controlled trials, and we ask them to have statistical significance and formal hypothesis testing in those 2 trials to demonstrate efficacy. But frankly, there are many, say, rare diseases out there. There are many situations where we have less than ideal data. We may have no good biomarker. And so despite all your efforts, you've gone sort of empirically into phase 3. Or it may be a rare disease that's highly heterogeneous. We have little understanding of natural history, and subsequent speakers are going to talk about this. And the patients are very precious. Okay, and so we can't afford to waste those patients. They're not that many, and their disease state is all going to be very different, and so forth. And so this situation, I think, is an ideal sort of test lab for where we need to extract the maximum amount of information from every patient about their response to the disease, because there are just not many people, and you can't do often 2 randomized controlled trials and get your answer that way. So we have evidenced a lot of flexibility in drug development because when it's a rare disease and so forth. So let me find my last slide here. Okay, here we go. So what are the barriers to moving forward? Okay, I only have 3 minutes. Okay, well, first of all, it's been hard. We've, of course, been encouraging developing and validating tools like animal models, biomarker qualification. We've come in for a lot of criticism lately on the biomarker qualification role, but really the thought, I think, is good, that we try to develop a mutually understood body of evidence about the performance of various biomarkers. The CAMD, which Dr. McClellan and others and FDA is involved in, the Coalition Against Major Diseases, has been working on better understanding of natural history by, say, doing an Alzheimer's model, other models, looking at the cofactors and the disease progression. But these things are very hard. It has not been easy. Drug and disease modeling, clinical trial simulations are increasingly being applied both in drug development and in our review. But again, these are not widely accepted. This has been mainly an ad hoc activity, and we have to think about how do we move this to a more routine part of drug development. So we probably need to think about the interaction in 1 minute with FDA and how to more formalize this early program. And this gets to the sort of final barrier I'd like to talk about in my 30 seconds remaining. The clinical medicine, okay, so let's move now here in drug development and back down in the weeds. Clinical medicine, the triumph of clinical medicine has been the randomized controlled trial and evidence-based medicine, right? And so that has advanced that field tremendously, and that is the gold standard, and that's what the clinical world relies upon, right? Okay, that is not modeling, that's not mechanistic data. So the problem with the clinical trial and randomized clinical trial, it's not really a problem, but you can only test a single hypothesis that way each time. And so there's you look at the population means, usually depending on how you constructed the endpoint, and you get an answer. It's a more definitive answer, okay, but you lose a vast amount of information about the patients in that trial and so forth. Of course, there isn't the same reliability to that information as you have with your null hypothesis that you were exploring, but you have lost a lot of information. And so I think the greatest barrier is going to be the tension between how much can we inform drug development with all these models and mechanistic data, and how can we make that reliable enough that then it feeds logically into that confirmatory phase 3 trial that is done, or trials that are done, and that we honor that information equally to the randomized controlled trial information, which is basically usually must be done because we can't foresee everything. But so we have the empirical tradition, which has been a triumph in medicine, okay? And we're trying to do something different here, which is the mechanistic scientific understanding that can lead to semi-reliable predictions. And I believe this tension, we need to bring it to the surface and talk about it, because I believe this underlies a lot of the failure to progress. I think that this dichotomy is seen in companies. I think it'll be seen in FDA, and I think you will see it in the clinical community as well. And to some extent, we're seeing that with the biosimilars programs that we're now trying to implement. So I wish you all good luck the next couple of days. I think this is a really timely topic, and I think the turnout shows how important this really is. Thank you very much.
Okay, great. Thank you, Dr. Woodcock. Our next speaker is Dr. Mark Rogi.
So I was told my mission was to inspire the audience and focus on these topics, and I guess I have about 13 minutes to do it here. So I am going to I'm going to go through these slides, rather fast, but I think we can get out, a lot of the main messages. In fact, a lot of the things I'm going to say are very consistent with what Dr. Woodcock has already mentioned. I want to start out, though, with something a little bit from my heart, and also where I believe we're going. I really am optimistic that there are a lot of new products that are going to be coming to bear. The productivity issue that we're facing is there, certainly. But with what we have learned, with this wealth of knowledge that we've gained over the last 20 years or so, is truly going to bring us a lot of new products for a lot of very serious unmet needs. You think the 1990s, we were unraveling the genome. And where we are today, we understand the human genome. We understand variability in that genome. We understand the regulation of DNA, through epigenetics. You think the HDAC inhibitors that are now making their way through development. We're modulating, DNA, essentially as a target. RNA, silencing RNA, altering the splicing of RNA. Those are new drug targets that we never would have been thinking about 20 years ago. And of course, our knowledge in proteomics, not only the targets, but the pathways and the networks and the larger networks, the interactomes, are just amazing in terms of what they can bring to us in terms of potential new drugs. On the flip side, the computational capability of where we're at. Again, 20 years ago, we were somewhat in the mode on the clinical pharmacology side of looking at exposures and looking at outcomes, and they were associations. Really, where we've moved since then, and I think we're going to be moving a lot further, is in the realm of more mechanistic pharmacology. We're going to understand receptor expression. We're going to understand how much drug is getting to that site of action. We're going to understand the variability in the response along the pathways, and then again, ultimately, the outcome. So, I want to say something here that I also really believe there really never has been big data. It's been a big myth. There's been these lag periods, these brief periods of time, really, when we just simply haven't been able to analyze all the data, the new data that are coming to us, and interpret those data. We're beyond that. I don't think there should be any daunting task here. We have the computational capabilities to understand at a molecular level, how a disease is occurring, and then in turn, how we might even be able to actually treat that disease. So what can we do? Within industry at least, to try to improve our productivity. I think there's three areas. I can't say an awful lot about capable leadership. That's going to be, an issue for the people above me at least to deal with, although I do have a slide in here that, hopefully won't make people too uncomfortable. Target validation. We do need to go, that extra mile. I think we have all been working with biomarkers for many, many years, and I see Carl, here in the audience, Carl Peck, and I think probably around 1996 you visited us at Biogen, and we were talking about how we could incorporate biomarkers into some of our programs. What we haven't always done, and I think this is industry-wide though, is really associate those changes in biomarkers with outcomes. That's that extra mile, and we'll talk a little bit more about that in some later slides. Sound dose regimen. As I've already said, we need to do a much better job, in really understanding at more of a mechanistic level what are the appropriate doses, dose regimens, the patient stratification based on receptor titer, variability in the signaling mechanisms underlying those receptors. We don't treat diseases so much anymore. We treat subtypes of diseases. We need to better understand those subtypes and then in turn appropriately dose patients on that. So just one comment slide here on leadership. This was a great study. It came out about a year ago. Mark Thuneke had studied companies that had sustainable and scalable R&D portfolios, and he found some common themes in various companies that were really able to sort of achieve this sustained success. And there were 7 traits, attributes, within these companies, common attributes. I've got 5 of them here that relate to what all of us are really involved here in the R&D world. But as you can expect, the complexity of what we're working in right now, the environment that we're working in, it requires an R&D pedigree. That hasn't always been the case in the past. Our R&D leadership now, our executive leadership, needs to have that understanding that just goes beyond a facile, capability to generally understand what we're talking about. Great products first. We need to understand the disease. We've got to understand the patient. We've got to understand that unmet need, and we need to create a molecule around that. And if we do it right, there will be a positive return on our investment. The next 2 comments are somewhat related: the courage to focus and strategic perseverance. No more jack of all trades and master of none. We need to have therapeutic expertise, something that in the past we haven't done. While we've maybe worked across 3 or 4 therapeutic areas, frankly, I think we each need to think, where do we want to be? Is it neurology? Is it immunology? Whatever. But don't go beyond that. We need to understand diagnostics, the competitive landscape. We need to understand the pathophysiology of the disease. We need to understand the patients and focus on that. And, of course, the perseverance. We can't dabble. We need to be in these areas and work in them for 10 years, 15, 20 years. It's not just the R&D excellence that comes from it. It's the operational excellence as well. And then finally, healthy disrespect for the impossible. We can't let anyone put barriers, around our creativity or what we believe the value can be in the technology that, we can get our hands on and utilize in the development of these new products. So, sort of taking this back now to the world that we live in research and development, what can we do in this clinical pharmacology, advanced modeling, and simulation realm to improve, productivity? Buckminster Fuller had it right. Don't try to take an existing model and soften it or change the shape of it around the edges. Throw it out, make it obsolete, and create your new model. Now, I think the good thing is, and you heard Dr. Woodcock talk about this a few minutes ago, in many respects, we're already there. We're already doing this. We're just not doing enough of it. And I think that's a little bit of where, the problem lies. William Gibson, I don't know if you've ever followed him or not, he said something that was very, very elegant, very simple, very true. The future's already here. It's just not very evenly distributed. We are there. I think we are there. We know it. We just need to bring the rest of our colleagues around with us, and they need to see some of these successes. Dr. Viglietta is going to be talking a little bit later about a relative success that we've had at Biogen with one of our Alzheimer's drugs, but it took a little bit of pulling along by some of us to make that trial happen and work. Now, what can we do to really improve that productivity? We do need to characterize validated targets. In 2009, Martin Whaling, who wrote an article in Nature Reviews Drug Discovery, actually talked about target validation. 6 years ago, that we are finding all of these targets, but we really don't know what they mean. I'd encourage people to read that article. I don't believe everything, agree with everything that he says in it, but it is certainly a very nice piece of work, and it should get us thinking about how will we validate our targets. Construction and qualification, applications of systems pharmacology models. We've done a great job of characterizing where drug goes in the body, what happens to it when it gets to various sites of action or sites of metabolism. We understand the sources of variability around how that happens. We need to probably do a little bit more work on the pharmacodynamic side, the pharmacology side. But overall, we're doing a great job there. Where we need to expand it further, though, is more on the mechanistic pharmacology side. We need to understand in a person with early disease versus late disease, or less severe versus more severe disease, how does that receptor titer change? Does the location of the receptor change from one tissue type to another? Or the patient, a 25-year-old male versus an 85-year-old female. We need to better understand from a mechanistic standpoint how much drug gets there, how much is necessary to get there, and then are there any sources of variability that we need to be aware of and consider to actually produce that outcome. For the sake of time, and I think to keep us a little bit on track here, I won't go too deep into this. There are many examples out there where some good target validation has occurred. We've seen this in the PCSK9 area. Some of our colleagues at Genentech here have been involved in modulating PCSK9 activity. It's a cofactor. It forms an adduct, with LDL receptor and traffics it essentially into a lysosome where it's degraded. In the absence of PCSK9, you'll get re-expression of that receptor. You'll essentially get lowering of cholesterol. A very nice mechanistic pharmacology study was published last year. It was followed up just this last month with a parallel population PK/PD study conducted by some of the very same people. And together that information informed the larger dose ranging studies, the dose regimens that were to be used potentially in different populations of people, whether they are on statins or not, to then conduct their later trials. So my last slide, and just two comments here on this. I do think that we are going to be seeing a lot of new products emerge and make their way to patients who really need them. I think the big issue is, what's that trajectory going to be? How are we going to improve the productivity, to get us to where we really need to be, frankly, to make our industry viable and keep it viable. And I think one of the key things that we need to do, really to make that happen, is for our organizations to recognize the value of systems biology. Is an area that is emerging in many of our companies. It needs even more attention. It needs to be utilized more broadly, and in parallel, working with systems pharmacology individuals, and for those of us in here who are already doing some of this, to do even more of it. And we need to interact with our colleagues on the research side in the computational biology area. So those are my comments. Thank you for your time, and I hope we can make this a very interactive and productive discussion. Thank you.